Great. Well, it's really great to be here and to get to share with you some of the things we've been doing in the recent years. My group at MIT and HHMI is devoted to developing and applying new tools to help us understand how the brain works. And so today I want to tell you three short stories about tools that we've developed that we think address certain aspects of understanding the brain that are difficult or sometimes even impossible to really probe with older tools. One technique I'll tell you about is called expansion microscopy, and it's a way of doing nanoimaging on ordinary microscopes. And I'll spend the first half of the talk on that, and the last half of the talk I'll split between the control and the readout of high-speed neural dynamics. Now, why do we build these tools? Why do we need neurotechnology in the first place? Well, the brain presents a number of challenges that I think are actually common to a lot of biological systems, but in the brain are particularly irreducible in the sense that you can't ignore this aspect of the complexity through an approximation in many cases. So one of the issues is spatial scale. Brain circuits are enormous. Brain cells, in the human brain, are centimeters in spatial extent, by far the biggest cells in the body. But if you care about synapses or ion channels or transmitters or connections between brain cells or the wiring of the brain, those often are nanoscale in dimension. And of course, the biological building blocks of life, like proteins and nucleic acids and lipids and sugars and so forth, are also all nanoscale. So the first half, I'll talk about a strategy that we've developed for doing nanoscale imaging across extended spatial scales. Now, the last half of the talk will be about sort of space's complement, time. Time is also really difficult in the brain because so many things in the brain require us to understand processes over a wide variety of timescales, all the way from milliseconds to, for electrical pulses and calcium events and spikes and synaptic events and so forth. But very often we need to understand these over extended lengths during the time course of a behavior, or in the cases of memory formation and Alzheimer's generation and aging and development, days to months to even years. So how do we build tools to help us cross space and time? Well, I'll start by talking about expansion microscopy. Many people have pioneered great ways of doing nanoimaging, electron microscopy, X-ray imaging, super resolution microscopy. There are all sorts of tricks for doing nanoimaging. But all of them struggle to be applied to large objects like brain circuits, and they also struggle in the sense that they often are slow or difficult to use. They require, for example, custom equipment to be acquired. In addition, it's difficult to see many colors. There are lots of biomolecules. Wouldn't it be nice to be able to see all of them in all of their glory? So starting about 8 years ago now, and then we published the first paper on this about 5 years ago, we decided, what if we could, instead of zooming in with a lens, we could physically magnify objects? And so we call this expansion microscopy. And basic strategy is to take a swellable polymer, such as shown in the cartoon on the left, and to chemically synthesize it so that these polymer threads will wind their way inside cells, outside cells, around biomolecules, between biomolecules, very densely and very evenly. And then, if we anchor the biomolecules or labels attached to them to the polymer, and we soften we soften the specimen, if we add water to the swallowable polymer and it expands, we should be able to expand the object. So let me walk you through the steps one by one, because I know this is a lot. Let me just point out that this technology owes a debt to many pioneers who came before us. In 1980, Toyoshi Tanaka, an MIT physicist, published a really important paper on polymer physics, where swellable polymers, when you add water, will undergo these very dramatic transitions. And so he was studying a charged polymer called sodium polyacrylate. This is better known as the stuff in baby diapers. Add water, it's drawn in through osmotic force, the polymer threads expand away from each other, and you get these enormous 1,000-fold volumetric increases in size. And that's facilitated by the highly charged polymer. Like charges repel, you get this gigantic swelling. That was published in 1980. Interestingly, around the same time, 1981, Peter Hausen and Christine Dreyer published this really cool paper where they were embedding specimens in hydrogels. They used an uncharged hydrogel, acrylamide. The charged version that I showed you in the previous slide is acrylate, subtly different but important. And they showed that you could embed different specimens in hydrogels and use that to facilitate immunostaining and imaging. So one might ask the question, could you take a cell like the one on the left and pull apart all the biomolecules until it looks like the cell on the right? Two biomolecules that are touching would now be some minimum distance apart, and two molecules that are some distance apart would now be scaled up by some linear factor. So the cell on the left would be expanded to look like the cell on the right, basically a constellation of biomolecules hovering in space with their relative organization preserved, but physically larger in an even way. Now, to make this work, we had to figure out several key tricks. Shown here in this cartoon are proteins in brown, and I'll start the animation here. The purplish blobs are anchors, handles, if you will, that we attach to the proteins. And so now we've developed handles that bind to DNA, RNA, proteins, and we've also worked out labels and handles that can bind to lipids, and now we're collaborating with Laura Kieslig's group at MIT to explore carbohydrates. To make a long story short, we need to attach these handles, shown in purple, so we have a way of pulling the biomolecules apart from each other. Next, we have to weave those dense meshes of swallowable polymer. And we do that by using a recipe not that different from what Hausen and Dreyer published in 1981. We bring in monomers, shown in little white spheres, and they undergo a process called polymerization, where they self-assemble into long threads of polymer. That's how we turn the monomer into the polymer. And when these polymer chains encounter a handle, they form a bond. And so if you think about it, the polymer threads are swellable. If we add water now, shouldn't it just expand? The polymers absorb the water, the handles bring the biomolecules along for the ride, right? But there's one more step we had to figure out. We have to soften it because things don't like being expanded. So we add detergents, enzymes, and or heat. And then when we add water, the sodium polyacrylate, the baby diaper material, absorbs the water. But this time, because of the handles, the biomolecules or labels attached to the biomolecules will come along for the ride. So we call this expansion microscopy. We first published this in 2015, showing the discovery that we could evenly expand a biological object. Fei Chen and Paul Tilburg were grad students in my group at the time. In panel B is a piece of mouse brain, and in panel C is the same piece of mouse brain about a day and a half later, and we expanded it 4.5-fold in each direction, about 100-fold in volume. And the polymer mesh, is shown in the upper left, it's very dense. The spacing is just a couple nanometers, smaller than many biomolecules. And when we add water, it swells so it looks like something in the lower left. Here's a movie. I hope it comes through the Zoom cast okay. Sped up about 50-fold. A piece of brain tissue we formed the swallowable polymer already in, and we add water about right there. And I hope what you can see is that this piece of brain tissue is swelling. Okay. So the first question I always get is the swelling even? And there are different ways of checking that. One way is to take a pre-expansion picture with a classical super resolution or nanoscale imaging method, and then take a post-expansion picture with a regular old microscope. That's what you see here in magenta, pre-expansion, and green, post-expansion. So to make a long story short, we've now done this analysis on many different tissue types, cell types, and species. The expansion is not perfect, but it's very good. We get distortions of about a few percent over a typical microscope field of view. And for the vast, vast majority of biological questions, that is fine. Another way to measure the expansion precision is to look at the resolution. Here are some cultured cells with microtubules immunostained with fluorescent antibodies. And we know what microtubules should look like thanks to decades of beautiful work by others. So we can take our image, deconvolve by the ground truth, and tell you how much error we have in terms of resolution error. I'll take a long story short, with a 300 nanometer diffraction limited lens and a 4.5-fold expansion, you might guess that the resolution would be at best 300 divided by 4.5, around 60 or 70 nanometers, and indeed, that's what we find. So that raises the question, if we expand more, Can we get even better images? And as I'll show you later, that's true. But even with 4.5-fold expansion, there are many things we can do that are difficult or even impossible to do with other methods. So here's a piece of mouse brain tissue that's pretty large. The scaled bar, well, you can see the cortex and the hippocampus. The left column is unexpanded, the right column is expanded. And we zoom in basically from top to bottom. Each white square is blown up into the picture below. The color code is shown on the left, where antibody staining with fluorescent antibodies. YFP is in green because some neurons are expressing YFP. And then bassoon is in blue and Homer 1A is in magenta. Bassoon and Homer 1A are pre- and postsynaptic proteins respectively. I hope you can see on the left, it gets blurry as we zoom in more and more, but on the right, we can see very crisply synapses. And that's zoomed in the lower right-hand corner, a pre- and postsynaptic compartment. And if we measure the distance between these pre- and postsynaptic compartments, as shown in the upper right, we get the same answer that Catherine Dulac and Xiaowei Zhuang measured many years ago using storm microscopy, a super res method that Xiaowei invented. But now we can do this imaging on a regular confocal microscope. So one way to look at this is that we're getting the resolution of a nanoscale resolution method, but the speed of a classical resolution method. And that's important because classical microscopes are getting faster all the time. I'll just give you an example here. Shown in the upper left is a piece of brain before we expand it, and the lower left is a piece of brain after we expand it. If you expand something 100 times in water, it's 99% water, and that means it's basically completely transparent. So that means we can do clever tricks like light sheet microscopy, where you shine light from one side and take a picture at a 90-degree angle. We were talking with Eric Betzig, who invented palm microscopy, another super resolution method, and he was originally extremely skeptical of expansion. But we decided, what do I know? I'm new to microscopy. He's one of the experts. Why don't we work together? And by the end of the project, we were working well together, and he had been a convert to expansion. So we ended up co-authoring a paper together where we expanded specimens and then imaged them on one of his light sheet microscopes, a so-called lattice light sheet microscope that their group invented. And so this is work that Rei Gao Shoisano and Gokul Padayulia spearheaded, working with many great collaborators shown in the lower right-hand corner. As you can see in these pictures, we can label mitochondria and lysosomes with antibodies against proteins found within them. That's shown in upper right in panel C. In the bottom panels, F, G, and H, we are antibody staining against myelin, an antibody against myelin basic protein. And to make a long story short, by expansion followed by light sheet imaging, we could go a couple orders of magnitude faster than competing nanoscale resolution methods. And it's probably only a matter of engineering to make it go even faster than that. In this paper, we speculated that with faster cameras and just a matter of engineering, different optics, maybe we could go 10 to 100,000 times faster. Here's the mouse cortex, layer 1 on the right, layer 6 on the left. Again, we're zooming in from top to bottom. Each dotted box is blown up in the picture below, and same color code as before. And we can zoom in from the thickness of the cortex, a millimeter thick, all the way to individual dendritic spines. So to summarize the first part of the talk, we've discovered that we can evenly expand a biological system, and this makes ordinary microscopes into nanoimaging devices. And it also means that if you use clever tricks like light sheet microscopy, you can do nanoimaging at blazingly fast speeds. So what can you do with this? Well, one interesting direction is, of course, to make maps of the brain. And so in a follow-on paper, we used a Brainbow, where you combinatorially express fluorescent proteins in neurons. So you see a bunch of proteins, a bunch of neurons at the bottom. Some are blue, some are pink, some are green, some are red. Each one's expressing a random combination of fluorophores delivered by random uptake of viruses. And at the top middle, you can see that we're zooming in. This is a piece of the mouse hippocampus in a region with dense axons. It's really hard to resolve individual axons. But in the upper right is the same field of view as in the middle top. And in the upper right, I hope you can see that we can cleanly resolve individual axon projections. So this is, of course, meaning that we can do brain mapping. Another interesting goal is to look at multiple kinds of biomolecules. So far, everything I've showed you is about proteins, but what about other kinds of biomolecules like RNA or DNA or lipids? So the next one we decided to tackle was RNA. This is work spearheaded by Oz Wassi and Fei Chen. And what we did was we could anchor proteins and RNA to the polymer and then expand. That's what you see here. The proteins, you can see some, for example, yellow fluorescent proteins shown in green, and then RNAs are shown in magenta. These have been imaged with post-expansion fluorescent in situ hybridization, or FISH, a pretty standard technique, although we do amplify the brightness using a trick called hybridization chain reaction. Anyway, to make a long story short, I hope what you can see here is that we can visualize with nanoscale precision, even in intact tissues, the location of individual RNA molecules at synapses. And this is very important, right? Neurons have one nucleus, but thousands of synapses. It's important to be able to visualize the location of individual RNAs to understand gene regulation. What about expansion factor? Everything I've showed you so far is expanded 4.5-fold or so, maybe 3-fold for some of the experiments, in linear dimension. Well, when J.B. Chang was in our group, and this is work now, we have a new version of this that Deblina Sarkar, Oswasi, Jinyan Kang, and Margaret Schroeder are working on. The basic idea is to expand an object over and over again. Take a piece of brain, polymerize it and expand it. That's what you see with the blue polymer in the upper left. And then the red polymer, we can form a second polymer in the space opened up by the first expansion and expand it again. So it's like PCR, but for space. We get exponential spatial amplification with a linear number of steps. And at the bottom is some chemistry that we use to transfer fluorophores, or more accurately, fluorophore targeting strands of DNA from the first polymer to the second. But as I mentioned, we have a better version of this working now. And so please drop us a line if you want to try this out. So with double expansion, what can you do? Well, the top half of the slide is done with storm microscopy by Xiaowei Zhuang's group, who kindly offered to help us. And one of the things that you can see with storm are microtubule sidewalls. They look like hollow tubes. And this is very difficult to see. Only a handful of techniques of any kind are good enough to let you see the microtubule sidewalls. But in the bottom, D, E, and F is double expansion. And I hope you can see here that we can resolve the microtubule sidewalls from each other. Here are some synapses. Panel A in the upper left is an excitatory synapse. Panel D in the lower left is an inhibitory synapse. If you recall, earlier pictures I showed you of synapses had pre- and postsynaptic densities touching. But with double expansion, you can see the synaptic cleft. And there you can see, for example, glutamate receptors or GABA receptors at these synapses. Here's brainbow again. Upper left unexpanded, upper right expanded once. At the bottom, expanded twice. These were done in collaboration with Dao and Kai, who is one of the people who worked on the Brainbow technology suite. And I hope what you can see here is that with double expansion, you can get very, very good resolution, even seeing very delicate structures like spine necks. So what do we know and what do we not know? Well, as I've told you, we've discovered we can expand objects. We can visualize different kinds of molecule by antibody staining against proteins or fluorescent in situ hybridization against RNA. But there are a couple of things, of course, that we don't know. So far, we've primarily validated our technique by comparing to known nanoscale resolution techniques. So getting down to resolutions below that, we don't know yet. So two proteins that are touching, can we resolve them from each other? That's below the resolution of even classical methods of nanoscale imaging with light. And so we haven't yet been able to validate that. But it's something we're working hard towards, is validating below the 10 to 20 nanometer resolution limit. But still, there's a lot you can learn by visualizing down to 10 to 20 nanometers. And in recent years, we've been working on applications of these techniques. When Yongshin Zhao and Octavia Bicure were working with our group and several pathology groups, Astrid Weins, Stu Schlitt, and Andy Beck, we showed that we could expand human tissues. So this is sort of a crowded slide, but on the left are non-cancer containing, and on the right are cancer containing tissues from top to bottom: prostate, lung, breast, and pancreas. The color code is shown in the upper right. But suffice it to say that for a wide variety of human tissues, we can visualize with nanoscale precision aspects of pathology. Limor Freifeld, when she was a postdoc in the group, we worked out that we could do expansion of the larval zebrafish brain, which is a very important species in neuroscience, working with a great set of collaborators shown in the lower right. Here are some synapses being visualized with cetatagmin and glycine receptors being imaged with antibody staining in the important sites of contact where spiral fiber neurons and M cells all interact. Jay Yu, who just finished his PhD in our group, working again with a great set of collaborators shown in the lower right, We showed that we could adapt expansion to C. elegans. So in summary, the expansion method can be applied to a wide variety of cell types, tissue types, and species. And I'll just end on this slide, which isn't really meant to be read. But the point I'm trying to make is that we think this is a very useful technique in everyday biology. Back before COVID-19, we did hands-on training of at least 400 research groups, I believe, and over 160 papers and preprints doing expansion-style protocols, not reviews and stuff, actual experimental papers have come out. It's being applied to look at protein synthesis and the nuclear pore complex and Drosophila neurons and dividing cells and stress granules and bacteria, and the list goes on and on and on. So if you want to learn expansion, please drop by our website, expansionmicroscopy.org, and we posted several protocols on that website. Unfortunately, we cannot host visitors to actually come and see us do it right now. But recently, we published a couple protocols papers that give very detailed step-by-step instructions, including photographs of how to handle the gels and so forth. So we hope that it's a fairly learnable strategy at this point. Okay, so the first half of the talk, I've explained to you that we discovered we could evenly expand biological systems. That lets you do nanoscale imaging on regular, ordinary microscopes, and you can use it to visualize proteins and RNAs and, in the works, other biomolecules in a diversity of cell types, tissue types, and organisms. But of course, as must be all too obvious, this only works in preserved tissues. I don't see how you could expand a fully living thing and have it still be fully living. So in the last half of the talk, I want to talk about time dynamics. How do we see and control biological systems when they are operating in real time? So I'll start with optogenetics, although I won't talk about it too much because most of the stuff is fairly old at this point. But in optogenetics, we control neural electrical activity with light. And so this started when Carl Dijsselroth and I were both students at the time, and we were just brainstorming, what are all the ways we can deliver energy to the brain in order to activate neurons? And we just started thinking about mechanical force and magnetic fields and light. And we thought light would be cool if you could get away with it because you could aim it at cells. And then you have to get light into the brain, and there are lots of ways of doing that, such as putting in optical fibers or more sophisticated optical devices. The next question then was, do we make a light receptor or do we find one? And it turns out that for about a half century, biochemists and microbiologists have been studying opsins, these 7 transmembrane proteins found in single-celled microbes. And you can see such a one in the lower left hand of the slide. That's bacteriorhodopsin, a light-driven proton pump found in single-cell organisms, where it helps convert solar energy into chemical energy. And then about a decade later, people found halorodopsins, light-driven chloride pumps from similar species of microbes. And then finally, around the turn of the millennium, several groups found light-driven ion channels, which they called channelrodopsins. So channels, of course, are not going to be useful for storing energy. They're going to dissipate energy down a gradient of voltage and so forth, and ionic concentration. And so these are used by microbes, in particular single-celled algae, to sense light to help them navigate. Anyway, to flash forward many years later, we now know that all 3 of these classes have members that can be used to control neural activity. On the left-hand side, light-driven proton pumps, as worked out by Xue Han and Brian Chow when they were working with me, found that they could be genetically expressed in neurons, and then you can shine yellow or green light on them. They will pump protons out of the cell and hyperpolarize them. And so one of these molecules, ARCH, has become quite popular for silencing neural activity. In the middle of the slide, halorodopsins, light-driven chloride pumps, Xue and also Amy Chuang, when she was a grad student in the group, found that certain halorodopsins could be expressed in neurons and used to hyperpolarize them and shut them down. So by deleting neural activity, you can try to figure out what a neuron is necessary for example. And then the right-hand column are channelrodopsins. The first of these that we put into neurons converts light into a channel opening that causes a flux of cations, in particular sodium and protons and hydrogen ions. And so that will then let you activate neurons. And so Carl Dijsselroth and I put the first one of these into neurons many years ago. And then Nathan Kopecky, Young-Koo Cho, Demian Park, and Amy Yang, and others in our group have been working to perfect these. So what do I mean by perfecting them? Well, in the previous slide, I kind of glossed over this, but each of these molecules is best driven by a certain color of light. So green light or yellow light are very popular for neural silencing, and blue light is very popular for neural activation. But when Amy Chuang was a grad student in our group for her thesis, she showed that we could actually develop better red light silencers. Why is that important? Well, red light can go deeper in the brain than other colors of visible light because it's less absorbed by blood. Blue, green, and yellow light are much more absorbed. So she found a halorhodopsin, a light-driven chloride pump that we named JAWS, which converts red light very efficiently into a chloride flux. And as you can see in the bottom part of the slide, we can then silence neurons 1, 2, or even 3 millimeters deep in the brain. And Leah Acker, who did her PhD working between Bob Desimont's group and my group, used this molecule and showed that for the first time she could get silencing of cortical neurons in non-human primates, because you need to silence a pretty big region in order to get the kinds of behavioral changes she was looking for. And the red light penetration to the brain, coupled to the pumping of chloride by JAWS, was helpful to her. What about activation? Well, when Nathan Klepecki was a grad student in the group, he worked on the flip side question: can we develop red light activators of neurons? And so we did a massive genomic search through over 1,000 plant genomes, over 120 algae, and found 61 new molecules, of which one of them had really good red photocurrents, which we named crimson. And as you can see in the lower left, you can use 735 nanometer light, which is like 100 nanometers redder than a red laser pointer, and still elicit spiking. And crimson is kind of cool as well, because we recently found out that it was advancing to the next stage of clinical trials for the treatment of blindness by a group in France. Nathan also found a molecule that we named Chronos, which is really, really fast. The advantage of doing a large, relatively unbiased screen. And Chronos and Crimson used together could allow you to control different populations of cells. Put Chronos in one population and activate them with blue light, Crimson into a second population, activate with red light, and then you can control two interacting sets of neurons. And now a couple of groups have actually used this to probe things like how do two sets of synapses onto a target neuron interact, or to look how one pathway in the brain modulates the power of another. Now, beyond two colors, how do you control many neurons? And we teamed up recently with Valentina Miliani's group, and their group is one of the pioneers of holographic multiphoton optogenetics. What that means is, like the microscopes shown in schematic form at the top, they basically sculpt light to make a hologram that is then projected onto the brain. And a hologram is basically a 3D sculpture of light for all practical purposes. You can see some fluorophores lighting up at the bottom of the slide. You see 6 different regions lighting up, and that's because the holographic stimulation projects light into that pattern. So Valeria Zampini and Dimitri Tanese from her group and Or Shemesh from my group teamed up, and we decided, what if we could make a molecule that would be matched to the power of these kinds of holographic stimulation methods? Now, one of the big problems, of course, is that if these molecules are everywhere on axons and dendrites, then you might aim light at one cell, shown by the dotted circle on the left, but you might hit axons and dendrites from neighboring cells. And so a trick that several groups, including Maclean Bolton's and Jawa Pan's and Frank Werblins, and at this point many others, has done, is to try to target molecules just to the cell body. And so the aforementioned groups took chenorodopsin 2, which is the original chenorodopsin that we put into neurons many years ago, and expressed it, fused to a small peptide, so that it would be what I like to call molecularly focused at the cell body. You know, we often talk about optical focusing, but can you focus a molecule so that it only exists at the cell body? Then when you aim your light, as shown by the dotted circle, it'll hit, axons and dendrites from neighboring neurons, but you're not going to excite them because only the cell of interest actually has any opsins to stimulate. So what we found was that a molecule, actually that had come up in Nathan's screen many years before, that we called COCHR, and it's very powerful, very high photocurrents. We found that we could fuse it to a small peptide and localize it to the cell body of neurons, which we call SOCOCHR. If we had known this molecule was going to be cool, we would have come up with a better name, but we didn't know that at the time. And you can see the difference between the left and the right images. The right images show hotspots at the cell body, whereas the left images show this the regular COCHR everywhere. In the upper right-hand corner of the slide, the stray excitation goes way down from 30% stray excitation to practically 0 in terms of stray spikes that we elicit. And in the lower right, because the COCHR molecule is so powerful, you can use brief pulses of light and get very precisely timed action potentials. So to summarize the second part, we were basically very, very lucky. These molecules from the natural world, from microbes, basically out of the box were sufficient to allow us to activate or shut down neural activity. They were fast enough, they were high amplitude enough, and they were safe enough. Which brings me to the last part of the talk. What about the inverse, watching neural dynamics? Well, the natural world has not made for us molecules that all by themselves light up when a neuron is active. Maybe an animal wandering around broadcasting its thoughts would be a bad thing in the first place. So we thought, this is work spearheaded by Erica Young and Kirill Pyakovich when they were postdocs in my group. We thought if the natural world won't evolve indicators for us that are fast enough, safe enough, and high amplitude enough, akin to how we lucked out for optogenetics, let's build a robot that'll do the evolution for us. And so the schematic here shows basically how we built an evolution robotic strategy. So how does this work? Well, directed evolution, of course, goes back many years before us. Francis Arnold and others won a Nobel Prize for their work in this recently. But we wanted to do multi-dimensional directed evolution. If you recall, we want these molecules to be safe, we want them to be fast, and we want them to be high amplitude. And if you evolve a molecule in only one direction, you might de-evolve it away from other goals. So here's what we did. We took a gene and we made lots of random mutants of it. Some are better and some are worse for any given goal. We put them into cells so that each cell, these are cultured mammalian cells, has just one copy of the gene mutant. Then in the bottom row, we use a robotic microscope to scan around and look for cells. And if we find a cell that has the right mutant, a mutant that's better for our goal, we bring in a robotic arm and suck out the cell and therefore the mutant, which we can then sequence, and the process can then be repeated. So each round, the mutants will be a little bit better for the goal for which we are selecting. So we did what at the time might have been one of the world's largest directed evolution screens done on mammalian cells, about 10 million mutants in 2 rounds of directed evolution. And what we decided to make was a fluorescent voltage indicator. We started with Quasar 2, which is a voltage indicator developed by Adam Cohen's group, and it's very, very fast. But it's not particularly bright, and it also had some trouble being localized to the cell membrane, but very pioneering for its time. And so we decided, could we screen for better localization to the cell membrane and better brightness? And on the right-hand side of the slide, you can see the multidimensionality of our screen. We want molecules that are better localized and more bright. If you're just one and not the other, it might not actually help you that much. But the fact that we're using a robotic microscope with robotic arms means that we can screen in multiple dimensions at once. In the case of the image on the right-hand side of the slide, we basically want to go down the diagonal. Now, this is a very crowded slide, but suffice it to say that the goals were met. In the upper left-hand corner, we got a molecule that we called Archon, which is really well localized in the membrane. In the upper right-hand side of the slide, it had very high change in fluorescence during action potential. You would shine red light and look at redder light with your fluorescence microscope. And in the lower right, you can see it's very photostable as well. We call this molecule Archon. And work collaboratively done with Shura Han's group at BU and others, Kirill Pyakovich, Seth Ben-Sussen, and Juan Sang recently showed that we could, as I told you earlier, we could make a soma-targeted version of this opsin. Sorry, it is an opsin actually, but we're not using it for its opsin function. We've removed that. Archon, I should say. And just as targeting a channelrhodopsin to the cell body helped clean up the background excitation, targeting Archon to the cell body helps clean up the background fluorescence. So by targeting Archon to the soma of neurons, we found that we could actually get what you see here, which are electrode-like looking traces from motor cortex, visual cortex, striatum, and hippocampus. And these are all taken with a fairly conventional 1-photon microscope as shown in the upper left-hand corner. We shine red light, again, 630 nanometer light, and we collect fluorescent light with a 660 long pass filter. We can look at populations of neurons. Here we are looking at about a dozen neurons. During the period of time that we imaged, 8 of these neurons had activity that we could detect. This is in the hippocampus of an awake behaving but head-fixed mouse. And because this is red light excited fluorescence, we can use it in conjunction with blue light driven optogenetics, and we can then drive spikes with blue light and image the resulting network activity with red light. And again, this is being done in the brain of an awake mouse. So recently, just a few weeks ago, we asked the question, or we published the result of asking the question, of whether we could use this targeting strategy, which I've showed you works for optogenetic controllers and voltage indicators, could we apply it also to other kinds of molecules? And of course, one of the most popular things to do in neuroscience in terms of neural activity imaging is calcium imaging, because calcium signals are very big, they're very robust in many cell types at least. And the modest kinetics actually means that they can be imaged on hardware that's not too expensive, amongst other wonderful properties of calcium imaging. And so we, this sort of line of thinking about molecular focusing got us thinking, what if we could put calcium indicators also at the cell body? If it's good for optogenetics by getting rid of stray excitation, if it's good for voltage imaging by getting rid of stray fluorescence, might it also be helpful for calcium indicators? So we took GCAMP6 and GCAMP7, and we screened a whole bunch of peptides. And we found, actually collaborating with Amy Keating's group, that a bioorthogonal coil peptide, these are human-designed peptides that don't really have any natural function in neurons, but by fusing those two GCAMPs, we could actually target them to the cell body. And so that's what you see here. There's a whole bunch of pictures on this slide, so maybe I'll just move to the next slide, which hones in on the most interesting candidates. So in panel A is regular old GCAMP6. In panel B is a version of the soma-targeted GCAMP where we fuse it to a peptide, but this one's kind of dim. And then panel C is where we use the coiled coils that we worked on with Amy Keating's group to target GCAMP to the cell body. And that one is just as bright as the regular GCAMP, as shown in panel I in the lower right. And so we call that soma GCAMP because we target GCAMP to the cell body. And I should note again that this was done all with a fantastic cast of collaborators shown in the lower right-hand corner of the slide. And Or Shemesh, Changyang Lingu, Kiro Pijakovic, Tripoli spearheaded this, again with the help of many, many collaborators. So what does it look like? Well, working with Misha Aaron's group, we expressed this in larval zebrafish. And I know this is sort of a crowded slide, but if you just focus on the lower left and lower right-hand corners, panel N and panel O, what we decided to measure was whether with a one-photon microscope method, which are, of course, very popular for calcium imaging because they're less expensive and faster than two-photon methods. But, of course, the sacrifice is that the one-photon methods have a lot more, out-of-focus light and so on and so forth. That's the price one pays. So we asked the question, with one-photon imaging, like through a light sheet microscope, will you get artifactual correlations between nearby cells? Two cells that are nearby, they might look similar to each other, but that might just be because stray light from one cell is being collected and assigned to the second cell. And we hypothesized that if we targeted these molecules to the cell body, we would reduce that correlation. And so if you look at panel O, just focus on the top half of panel O, with GCAMP, what we're plotting here is correlation on the y-axis and cell-cell distance on the x-axis. I'll point with my mouse because it's a really crowded slide. And with a soma-targeted GCAMP, we showed that we actually could bring those correlations down. Now, people have been using nuclear GCAMP for a while. That's another way of getting GCAMP to be away from the axons and dendrites, but you pay a price of speed. But we showed that with soma GCAMP, we can get much lower neuron-neuron correlations, and we can actually approach what people get with nuclear-targeted GCAMP. Working with Xue Han and Kate Tai's groups, we looked at mammalian brain in the live mouse brain. And we again showed that we could use, this is work in the Han lab with the mouse striatum, comparing regular GCAMP and soma GCAMP. And again, if you just look at panels J and K, the GCAMP, this is again correlation plotted versus distance, we get much lower correlations with soma GCAMP than we do with GCAMP. So that's really nice. And then here's some work taken with an endoscope from Kate Tai's group, where again, we show that we get better quality data with the soma-targeted GCAMP than with the untargeted GCAMP. In this case, our field of view is fairly small, so we, as we showed in the supplementary material, the correlation analysis wasn't too meaningful, but we did see improved signal-to-noise ratio, what we think is a more accurate spike count, and other effects of cleaning up the background fluorescence. So to summarize the last part of the talk, we really want to try to do for neural imaging what for optogenetics we were just lucky, improve the signal, improve the speed, improve the safety, and so forth. And to do that, we built robotic directed evolution strategies. We've used that to help develop new voltage indicators, but more and more we're thinking about how to use these indicators. And so a theme that pops up several times in the last half of this talk is what you might call molecular focusing. This idea that you need more than just the focusing of light, you need to focus where the molecules are. And I've shown you 3 examples where different teams within our group have done this for optogenic controllers, voltage indicators, and now calcium indicators. So all these tools we share freely. The molecules are at or on their way to adgene.org, for example. You can go to our website, synthneuro.org, synthnero.org, and it'll contain links to protocols and where to get DNA if you want to try things out. I'm very excited about how we might combine these technologies. What if you could look at the neural activity, perturb it, and then map molecules, wiring, connections? Could that be useful for more comprehensive descriptions of how neural circuits work? And so just as a pointer to the future, in our group, we're starting to do some science as well. And it's early days, but we're very excited about ways of combining these tools or developing new tools that help us integrate different kinds of data in order to understand neural circuit functionality. So I think I'll end there. Along the way, I hope I've mentioned the leaders of the projects within our group, as well as key collaborators. But like anything else in neuroscience, this is a large interdisciplinary collaboration. At the top are people in the group who've helped with these projects and are co-authors on papers. And in the middle is an even longer list of collaborators all over the world who we work with. And again, stop by our website, synthneuro.org. We're always happy to share tools, and hopefully we can help you solve something of interest to you. So I think at that point, I will take questions.